So first of all, just to reiterate what Grant already said, the same applies, right? You need to take into consideration soil variability and topography and previous management. One big difference, though, is that for most physical properties that you would actually take a soil sample from and send to a lab, you really want to focus on that surface soil area. And so no deeper than 6 inches. In fact, in my research, we often will sample the top 2 or 3 inches only. And the reason is that biological activity primarily happens in those top inches, right? Biological activity doesn't disappear, but it drops very rapidly as you go down to the soil surface. And the majority of that biological activity happens near the surface, and that's where microorganisms are breaking down organic matter and affecting physical properties. And so just to give you a couple of examples, aggregate stability might be one test you're interested in, or you might measure water infiltration. Biological properties you might be interested in might be soil respiration or mineralizable nitrogen, microbial biomass, or even microbial community analysis, various techniques for looking at that. And all of those kinds of tests, you want to limit your sample to the surface. Now, if you send it into a lab, as Grant said, they'll probably give you specific directions on the specific depth that they want. So make sure you double check that. The other really important consideration with physical and biological soil properties is soil moisture. For soil chemical property measurement, you pretty much air dry the sample, or the lab does for you, and everything is reported on a dry weight basis. The problem with, or the unique challenge with measuring physical and biological properties is that a lot of them are dependent on soil moisture status. And so, for example, if one year you take your samples right before an irrigation event, and then the next year you go in and take your samples right after an irrigation event, you may get very, very different results back from the lab and be totally confused. And so the way we get around this in irrigated systems is easy, right? You just make a habit of taking your samples 1 or 2 days after irrigation, and then you know that you've got pretty consistent soil moisture conditions from between sampling periods. In dryland area, areas that's more challenging, and in that case, watch the weather, take your samples in the spring, and try and choose a time where there's a consistent soil moisture year to year, not too wet, not too dry. So what I really want to focus on today, though, is this question of, well, do I want to send my soils off to a lab for biological and physical testing, or do I want to consider field measurements? And I'm sure Matt is going to touch on this to a greater extent, but I just wanted to pull this up, right? There's very few commercial labs that do soil health testing, but that is changing, and the Cornell Soil Health Testing Facility is one of the well-known ones. And this is just the list of tests that they offer under that. Though even this, there's actually very few biological, right? They've got wet aggregate stability there. They've got soil respiration, total carbon nitrogen, pretty standard. This autoclave citrate extractable ACE protein test is correlated with nitrogen availability. And so most of these are really pretty limited in terms of what they can tell you about your biological and physical properties of your soil, simply because they are chosen for their ease of commercial lab use, right? A commercial testing lab has to be able to test something that's relatively quick and easy and can be managed with a large throughput, right? And so it's still limited available commercially. And so there's a real strong argument to be made for learning and considering taking your own on-farm soil health tests. So what would that look like? Well, I had a graduate student start to answer this question just a few years ago, and she immediately got totally confused. We ordered the NRCS soil testing kit. It's a massive box, and you get 1 million different little pieces in it, and it's really overwhelming unless you're a trained professional, right? A soil scientist. And the NRCS, of course, is very willing and able to help you and come out and train you in these tests, but still there's quite a learning curve. There's various sorts of at-home rapid nutrient testing kits available, but the question is, do they work? There's also various visual assessments that you can do, like this one here in the bottom right, excuse me, that's a, it helps you score your soil against some kind of a card with photos. And then the Solvita soil respiration test is widely available and for measuring soil respiration, and that actually works very well, but it's also pricey. So what do you do? What do you recommend to growers? And this is what the question that we decided we wanted to try and answer. And so my student and I, we got some dollars to start to look at this. And she, like I said, we ordered the NRCS soil testing kit. We ordered a bunch, we basically went shopping and got whatever we could get and laid them all out and then kind of ranked them. She tried them out, ease of use, cost, that kind of thing, time. And then we selected a few laboratory tests to compare them to, right? Because we wanted to know, okay, this test might be simple and easy to use, but is it accurate? Are the results meaningful? Do these simple tests differentiate between land management systems out on the farm? And that was really the goal. If you're interested in reading the paper, the reference is there. It's open source, open access paper, so you can download that and read that. We also have a fact sheet summarizing the main conclusions that we came to. I will say that we tested this in an orchard setting. However, this does not mean this isn't relevant to other types of farming. Field crops, for example. It's just that I was working with orchards at that time and I had some other funding, and it made sense to couple this project with the orchard work we were doing. And so we had a bunch of different treatments in these orchards, randomized complete plot designs with 4 reps of each. And so we had, at that point, like 4 years of documented soil health differences across these different treatments. We had everything from conventional fertilization with herbicides, all the way to organic with mulches, living mulches in the tree row, right? And so you can, and this picture on your right is actually a tillage strip. So that was organic with tillage. So we had organic with living mulch, organic with tillage, conventional with compost, conventional just with herbicide, conventional with mulch, organic with mulch. It was fun. So what did we discover? Well, we focused in on this one particular test, and I'll show you how you do it in a minute, but we called it the 7-bucket Slake test. Because when my student was trying out the Slake test, is in the NRCS test kit, is this you basically do it on a single aggregate, right? A tiny little piece of soil with a little sieve and a little dish. And it was just really picky to work with. And it was actually Bob Newhall who suggested, well, why don't we just scale this up, right? Make it big, make it easy to do, and maybe it will work better. And so that was one of the tests that we tried and turned out to be the best one of the bunch. And so here's just some data showing this. So percent stable aggregates on your y-axis here is basically just a visual estimation of the stable aggregates remaining after performing this procedure. And then we correlated that with microbial biomass in the soil measured in the lab. And you can see these are really strong correlations. They're over 80%, and really nice differentiation between our different treatments. So the blue triangles here, this is the conventional with herbicide. The yellow here was the tillage organic, orange squares, straw mulch organic, and the gray triangles were a living mulch organic treatment. 2 years of data, really nice treatment differentiation. So this particular test was really good at being able to differentiate soils with known soil health differences. So what did that look like? Well, it looked just like this. We got a kitchen sieve, we just got a shovel and filled it up with soil. We just, limited the shovelful to the top 4 or 5 inches, filled that up, and then lowered it and raised it in that bucket. I think it was 5 times. And then we photographed the results. And so now I just want to give you some pictures because you can just see how dramatic the differences were. So these were 2 soils. They were both silt loam soils or loam soils. And this one here on your left came from a peach orchard that had been tilled for 30 years, right? And you can see there's virtually no aggregate stability left in that soil whatsoever. You've got a shiny surface, right? And really nothing much to score there. Compare that to the same soil textural class, right? The same loam soil, different location. This one had been under grass for 30 years and had just been broken out into an orchard. And this, believe it or not, had the same treatment as that one, but you can't even tell it's been sitting in a bucket of water, right? It just shed the water. It almost has a waxy quality, and those aggregates did not degrade at all. So those were the extremes. What about our orchard soil? So these both come from the Kaysville orchard, those pictures that I just showed you. And the picture on your left is one of those organic treatments. This was the one with straw mulch. And this one on the right was our conventional herbicide treatment. And all these samples were taken in the planting in the strip, right? And so you could see that even between those 2 very relatively similar treatments, just been in place for 4 years at that point, you could still differentiate the 2. Now, what about other biological tests? So that was a physical property. It was closely correlated with microbial biomass in the soil. This was another one that was actually pretty good. We did the typical worm count where you dig a pit and you count all the earthworms in it, and that one's recommended by the NRCS. We found that, that was hard to work with. There was just a lot of variability. This was a variation on that actually Diane Alston helped us come up with. It was more like a Burley's funnel test. adapted to the field. So we took our soil sample, we put it in a funnel, and we left it out in the sun with the bottom of the funnel in a glass jar, and all the insects that were in that soil fell down into the water, and we could count them. And so this was the results from that. Pretty good correlation between, in this case, soil respiration. Our top tests and were these here. So that sieve and bucket test I showed you. There was a variation of that one called the hose test, where we sprayed the soil with a hose for a set number of minutes under certain pressure. The total organism count and the Solvita test, I showed you a picture of that earlier. Those were the ones that correlated to our lab results most consistently and accurately. And so we took those out into the field, tested those with growers on growers' farms. We did this one-on-one with some of our local growers, and we also presented this at a number of field days. And we asked them, what do you like, right? And hands down, people really, really liked this sieve and bucket test. And that link at the bottom is the link to the fact sheet that's up on the USU Digital Commons. You can also find it through the Production Hort website that basically describes in detail how we did these tests. Now, I don't think this is my last slide. I don't think this presentation would be complete without mentioning this new smartphone app that's really getting at the same idea. It's called Slake. Was developed a few years ago by a lab in Australia. And it's just basically a smartphone application. You put your little bit of soil in a petri dish, and then you run this, and it takes pictures of it. Basically, it measures how quickly those little pieces of soil dissolve in the water. And so it measures the increasing bands or zones of cloudiness around that little lump of soil. This is a recently published paper that also did something similar to us, right? They compared this test to various other ways of measuring aggregate stability, and it won hands down. And I should also say that there's a reason that we correlated our test to microbial biomass or soil respiration and not lab aggregate stability. For some reason, measuring aggregate stability in the lab is the biggest headache, and it's not the most predictive test at all. It's actually quite problematic. We have tried this though, and in our soils, in Utah soils, it doesn't seem to work so well. And in this particular paper, they measured this on high clay soils. Our soils tend to be silty, even sandy. We just don't have that structure to start out with. And so basically what was happening is that our soil heads were dissolving way too quickly for this thing to really capture any treatment differences. It was just giving us poor results all over, right? But this was out at Snowville in a tilled situation. So we were definitely putting it through its paces.